then introduce. Okay. Welcome to the DDPS seminar, everyone. Before we introduce our invited speaker today, let's go over some rules and logistics. First of all, please mute yourself during the talk, but if you have questions, you can unmute and ask questions, or use chat room to post your questions so that we can address them in Q&A session at the end. Second, today's DDPS seminar is open to external audiences. Therefore, no classified discussion is allowed. Finally, the talk today will be recorded and uploaded in our YouTube channel. That's about it. Now, let me briefly introduce our speaker today. It is an honor to host Dr. Garikipati, who is a professor in the departments of Mechanical Engineering and Mathematics at the University of Michigan. He obtained his PhD at Stanford University in 1996. Between 2016 and 2022, he served as the director of the Michigan Institute for Computational Discovery and Engineering. His research is in computational science with applications drawn from biophysics, material physics, mechanics, and mathematical biology. Of recent interest are, of course, data-driven approaches to computational science. Today, Krishna will present an interesting topic, which is Fokker-Planck-Inverse reinforcement learning. I'm sure we will hear an exciting talk today. But before I pass the baton to Krishna, let me ask a couple of questions to Krishna just to get to know you better and make this seminar somewhat informal. Nothing serious, okay? So don't worry. First question goes, obviously, you're passionate about computational science to the applications to biology and the material science. Is there any moment in your life when you realize that, wow, this is it? This is the field I would like to commit my career to this. If you want to share that.
So I do remember when I think I was a postdoc when I decided that, I remember a particular moment when I said, yes, actually the materials physics work was really interesting. And I remember that happening. And that was during my postdoc time. Then I came to Michigan, and I remember some of my colleagues were already working in not quite biology, but really soft tissue, doing soft tissue mechanics and so forth. And there are many things we can do from computation there. So this was early 2000s, and I remember telling them at that time that yes, bio is very interesting, but I think I have my plate full. But then I was looking at some problems in growth, just growth of continuing growth, right? And the application of biology is very obvious. And then I realized that, well, here I have this. If I don't use it, I lose it. So even though I thought my plate was too full, I started looking at problems in biology, and that has grown. I went and got the problems. I went into cell mechanics, and I looked at embryogenesis and things like that. And then, of course, when the interest in scientific machine learning came up, it was very natural. I think most computational people find this to be a natural extension of the work we've been doing in computation. So yes, we continue to draw applications from biophysics as well as materials physics, and but it's very clear I'm in some cases I'm considered a computational materials physicist. Some people consider me a computational biophysicist. But I think the computation comes first.
I see.
Sorry, long-winded answer, but hopefully that's it.
No, very good, very good. I guess you chose a right field to commit your career to. I mean, because of that, you got many awards during your career, and one of them is Presidential Early Career Award for scientists and engineers. That seems very special. Would you mind sharing some story about receiving that award? And
yeah, actually, that award has the PCASE, as it's called, is something that all the agencies can put forward nominees for across all agencies. At that time, it was actually there's a good connection with the labs, because I was advanced as a nominee through the DOE. And so I have colleagues, and I've been working with people at Sandia for a long time, Sandia, California, just across the road from you guys. And that was the nomination. So yeah, so it's pretty special, and that was a long time ago.
Yeah.
But that was for it was of course early career, and
Right.
that was when I was still just thinking of some of the ideas from a long time ago that step by step brought me here. So
Incremental.
yeah, incremental, definitely.
Awesome. Well, thank you so
No,
much.
thanks for the question. That's nice.
And we are looking forward to your exciting talk.
Great.
The floor is yours.
Thank you so much, Yamsu. Well, good morning. Well, I shouldn't say, I know people are joining from all over, so whatever, good day to everybody, wherever you are and all that. Right, so I guess those remarks and that conversation gives some context for this work. You can see the title. It has a number of co-authors, and I'll talk about them at the end of it, so I can really, give you even faces to the names. All right, so let's start. So the problems, like I suggested, that we work on have been sort of inspired by biology, but of course they hopefully have wider applications. In this particular case, we are basically looking at cell dynamics, right? Cells moving around. Something that's known. So I'm doing this in PDF, so the cursor will change. I think you've got a clock right now, watch right now, because there is a movie running. But anyway, so something that's known is that cell dynamics, being populations moving around, obeys at some level it obeys the physics of transport problems. So what we're seeing here, the black and white movie that you're seeing here, is a type of breast cancer cell. As you see the designation there, this is a breast cancer cell line. It's known to be very aggressive. It aggressively moves. Motility refers to movement, and proliferates also. The particular experiment we're looking at here is what is called a scratch assay. You see the schematic here. You take a monolayer of cells, you literally scratch it, right? You actually introduce a cut in it, and then you sit back and watch what happens. And what happens is that typically these cells will try to close the wound. It's a wound healing process. That scratch that we introduce, the cells will kind of move into it and try to fill it out. That's the movie that you're seeing here. It loops over. So when it starts now, you see the black space in between. That is the scratch. And you see the cells coming in from both sides. Now, if you just stare at this movie for a little time and you say, okay, what are the cells doing? Something natural to think is that they're moving into the scratch. You can see them moving into the scratch. And so one may say that maybe there's some sort of directed motion. And that directed motion is suggested by this cartoon here to the left. But then if you look at it for a longer time, you may say, hang on, I think there's also some sort of random walk going on, right? Because they're moving in one direction, but maybe they're also moving in all directions to some extent, and maybe there's a superposition of this. So the middle cartoon here is meant to indicate a random walk, so essentially a diffusive type of process. And finally, if you look at if you look at certain of these cells, they become bright, that one. So it became bright for an instant, and then it divided. So the cells are doing this also, right? So a mother cell gives rise to daughter cells. Now, if you step back and think about this as a transport problem, and you say, okay, I'm not going to look at every single cell. I'm going to define like a population. I can define a cell density. You can write out one of these types of equations, right? Essentially, it's an advection diffusion reaction equation. So the directed motion is advection, the random walk is diffusion, and this proliferation of cells, or even if they die, is a reaction term. Okay, so that's the sort of, and so essentially this can be, you can subject this to a PDE model. The movie on the bottom is also from a scratch assay. It's a slightly bigger we're stepping back and taking a view of this. Each of these little the pixels that you see is actually a well. And if you can see the dimensions here, right? So this is 3000 microns, 3 millimeters, which is actually pretty big for cells. So inside each of those are cells, and the colors you're seeing are cell densities, and the same thing's happening, right? We've introduced a scratch around the middle in the vertical, and the cells are moving. So, sorry, was there a question? Okay, and the cells are moving in, right? So there's just a different view of the same sort of thing. So one can describe this type of motion, this type of dynamics of cells through PDEs. So you see the hypothesis up there. What I'm doing here is taking that second movie and said that, well, if you look at this, there's really not much interesting going on in the horizontal direction. So maybe we can just average over the horizontal direction, or maybe just look at what is happening along one particular vertical line. Okay, so if you do that, then you can sort of collate these data in a different way. a way to show such time-evolving data in biology is what is called a kymograph. It just means that you plot something, in this case, the y position along the vertical axis, and along the horizontal axis, you put time. Okay. So now let's understand what is happening here. So what we're plotting here are the cell densities. And if you look at the early times, you see there's a large region in the middle where the cell density is low, the blue colors. And then as you go along in time, that middle region fills up as the cells come closer together and they get to hotter colors. So now, okay, so and then the dotted lines here are just tracing out contours, right? Contours of constant concentration or constant density. It turns out that if you look at the form of some of these contours, they have this form y equals something time to the power b. That exponent b is actually of the order of about 0.4. And so that suggests, oh, it's 0.4. That's almost 0.5, right? So is diffusion dominating, right? It's a natural sort of question when we look at this kind of a plot. So what we are aiming to do then, what we aim to do is to say, okay, can we actually become more quantitative about this? And this is what we did, right? So we looked at cell dynamics. We said, okay, we have this advection diffusion reaction approach. Let's make some ansatz for all the parameters. We're saying that diffusion must be positive, right? So we say diffusivity can depend upon in this fashion, and we allow for the possibility that everything can be even positionally dependent, right? So we expand it as a polynomial in y. Y tilde, which is a scale, so that it goes from, what is it? It goes from -1, -half to half, right? So, or something like that, right? So we say that diffusivity has to be positive, and all these parameters, the thetas also have to be positive. We say the direction velocity is 0 at the midpoint because cells are moving up or down. So we write in it, we write this sort of an ansatz with the effective velocity. We also write the reaction as a polynomial, right? All as functions of position. Also, we make the theta parameters time dependent. So we're allowing all these coefficients to be basically spatiotemporally varying. Okay, so we say, okay, this is my ansatz, right? Because I want to see how these things are changing. You know, the cell dynamics may not be constant over time and space, so I'll let all these parameters vary in space and time in this fashion. So if I write this ansatz, okay, so we can write this ansatz and we can say that, well, I have the data. Can I infer what's going on, right? Can I infer what these coefficients are? That's what we aim to do, and we aim to do this in a sort of somewhat principled manner. So we look for parsimonious model selection. The method we use, there are many methods to do this. The method we use, we call it variation system identification. It has some relation to these other algorithms called SINDI, sparse identification of of dynamics. The difference is that we do everything in weak form, right? So we take that transport equation. It's written here as a time rate of some cell concentration. And here, chi indicates any operator, right? Maybe a diffusion operator, maybe a reaction operator, advection operator, and so on. The theta is the coefficient multiplying it, right? Linear coefficient. We do everything in weak form. So this is what diffusion looks like. That's what advection looks like. That's what reaction looks like, and so on. Now, from weak form, of course, you can go to a finite element description of this in a matrix vector form. A lot of our computation is finite element based. So that's what we do. If you write out a finite element residual for this, you go through the usual steps, which may be familiar to many of you here. The A here is the assembly operator, R is the residual, which typically in a finite element setting, we demand that the residual goes to 0, and we look for a forward solution. Here, however, we are saying that, look, we have the data, we know the solution, we need to determine the coefficients. We want to determine these coefficients theta. Theta is now a vector of all the coefficients multiplying all our operators. Essentially, this is an optimization problem. We say that the theta is the argmin of this residual, right? So we are trying to minimize this residual. It's a squared residual, so minimizing it means sort of the residual going to 0, and we introduce some penalty regularization so that the parameters don't get very big. Now there are many parts, and then your aim is to find the argmin. There are many parsimony promoting approaches. You can, the one that we use is called stepwise regression. I won't get into details about it. I can talk about it. There are genetic algorithms and so on. Essentially, it means that you choose parameters, you find an argmin, and you find a way to eliminate parameters until you come to some parsimonious set, balancing representation and parsimony. Okay, this is what we do. So we've written a few papers on this, and you see a couple of early references. There is more work that's been done since then by us. Okay. we also do this sort of parameter refinement, which is to say that, okay, we use this approach to decide which of these operators are on or off, right? You choose them. And then once you do that, you say, well, I may want to refine the actual parameters of the operators that are active. And you can do things like PD constrained optimization after that, right? It's fairly standard stuff. We use adjoint-based methods and so forth. So this is pretty standard stuff. Okay, so what does this all tell us? Here's what we see. Okay, I'm going to step you through these plots. The first one on the left here, if you can see my hand cursor, it's the original data for y versus time, this schemograph. We inferred a model, and then we ran the forward model after inferring it. This is the result we got. It's fairly similar. There are some differences. One can look at where the error is. The error has, there are some hotspots where the error is high, but it's all of the order of, if I'm not mistaken, this is of the order of percent. Yeah, that's percent error. Okay, so there are some hotspots. But what does it tell us, right? So we do infer a diffusion, and remember the diffusion, all the parameters we infer are spatiotemporal. So what we're seeing is that versus position and time, this is what the diffusivity looks like, right? Mostly some low value of diffusivity, about 0.2 square microns per micron square per second, and there are some hotspots, right? There are some points where we picked up bursts of activity. We look at advection, so we're looking at the advection velocity, how much advection there is, but I'm presenting it in terms of the Peclet number, right? So non-dimensionalized with respect to diffusion. What we're seeing is that it's mostly zero, mostly this green region, right? Mostly zero, and then wherever there are bursts of other activity, we do pick up some bursts of advection, but mostly zero, right? Finally, the reaction we show in terms of the Darmkeuler number, it's very small. The Darmkeuler number is essentially of order 10 to the minus 3. So it's suggesting that proliferation plays a very small role here. There is some directed motion, yes, but mostly it is diffusion, right? So what we saw originally in these data, where if you look at these contours of constant density, you may say, well, that looks like T to the half, right? Which would be classical diffusion. We're getting something very close to diffusion. We're actually inferring all these parameters and seeing that, yes, diffusion does dominate. So that suggestion early on that we're getting T to the maybe 0.4 is reasonable. But this is, again, a principled way of trying to really figure out what the cells are doing. So we've written some papers on this. There is presently an archive preprint which describes our methods to look at, to infer these types of reaction, advection, diffusion models for wound healing. Okay. Okay, but where is this going? So this is good. You can have this sort of mean field approach to looking at cells. Sorry, going the wrong way. Okay. But then we started looking at other problems also, right? So we took the same cell line. Okay, sorry, I'm being klutzy here. Sorry about this. We took the same cell line here, and we now put a chemical gradient upon them. Okay. We put a chemical gradient where the gradient is sort of attracting the cells to go leftward. It's the same cell line, but you see here the cells are a little more sparse. The two movies that you're seeing are essentially the same dynamics, exactly the same things going on. On the left, you're seeing only the nuclei. On the right, you're also seeing the cells. Okay. Now, this chemical gradient has been put, and the cells are being attracted to it. This is a chemoattractant. It's called CXCL12 here. It's this type of cells are supposed to move in the direction of that chemo gradient. Now, if you look at these cells, you will see that, yes, many of them are doing that, right? So you see the cell tracks. This guy is moving to the left, moving to the left, they're moving to the left, but not all of them. Like you look at the one at the bottom here, right? It started out in the movie. You see it's moving slightly to the right and then sort of changes its mind, gets confused. And, if you look at the one up here, near the top right here, it is definitely moving rightward. This guy up here is a little more uncertain about what it wants to do, but so on, right? So the whole point is, and this is long-term data, this is like 24 hours of data. So the whole point is that there is a lot of heterogeneity in cell response. And while one may think that this is just stochasticity, there is a strong suggestion in cancer biology, that the heterogeneity of cell response is not just stochastic, it's actually systematic. It's a significant element of progression in cancer. And the reason heterogeneity becomes important is that you may have a treatment that is targeting a certain type of activity of the cells. But because of this heterogeneous behavior, some of the cells escape. And then there is other evolutionary things come in. Those cells are which can escape whatever you're doing to them because they're doing something different. They're reacting differently. They are the ones that survive. They outcompete the others. They become stronger. They get more resources. And that is one of the things that makes these types of diseases hard to treat. Okay. Okay. So how do we approach this problem? So first of all, we took this cell data and we did a k-means clustering on them, right? To see, okay, what type of behavior are we seeing? We found 3 clusters to dominate. So you can see the binning and all of that. This is the loss of the k-means clustering. Three cell clusters. Cluster 0 is and as you're seeing all the cell tracks here. Cluster 0 are cells that were moving leftward, right? And just like the chemoattractant was trying to do, trying to make them do, right? Wanted to make, it was put to make them move leftward. But then there is this renegade contrarian group of cells which is moving rightward, right? And I pointed out some of them. And then there's a third cluster which is kind of not quite sure, moving slightly leftward, but maybe a little more unsure about what they're doing. Okay. So we see these 3 populations. So what can we do with these 3 populations? We could apply our mean field approaches to these populations, but there is a difficulty. There is a difficulty in that when you are, if you try to apply a mean field approach to the whole population, the fact that there is so much heterogeneity in them, there is distinct heterogeneity, means that your mean field approach, first of all, does not make sense. You need to have more populations. So then you may say, okay, I'm going to apply my approach to each of these populations, right? Each of these clusters. You can do that. I hope you can see this on your screens. What you should be seeing is that these are all cells, okay? So every dot is a cell. All the cells are represented. The one on the left is the cluster that's moving leftward. So what we've done is of all the cells, we've put in red the cells that are moving leftward. Okay, so you can see if you stare at the movie on the left, you will see that, yes, on average, the red cells are moving leftward. If you look at this cluster in the middle, you will see that, again, on average, the cells that have been highlighted red in this movie are the ones moving rightward. And the third cluster, you can't quite tell, right? Are they moving leftward, rightward, and what so on. Now, already it turns out that actually these populations, each of these subpopulations are getting very sparse. So mean field approaches are already beginning to get shaky. But what we did was we took every cell, we put a Gaussian on it, kind of smoothed out, got some smoothed out density. Those are the contours that you may be able to see just evolving in the back of these movies. So we did that to be able to infer something, right? Though we know already it's shaky because we don't even have enough density here, right? So totally each of these groups, for instance, are of the order of about 300 cells. And as I recall, the whole region in which they are moving is of the order of a few hundreds of microns. Yeah, maybe even 1,000 microns. So it turns out that the cell density is not very hard to do this. But anyway, we can go ahead and do this. So what do? We do, again, this advection diffusion model. What we find is for the 3 clusters, 0, 1, and 2, we listed the diffusivities. You see them here. Sorry. Oops. Going the wrong way. Okay. We also inferred the advection velocities. So what we are picking up correctly is that this left cluster is dominated by leftward movement. E1, E2 are just the basis vectors, right? E1 is in the X direction, E2 is in the Y direction. So this 0 cluster is indeed moving mainly in the leftward direction with some amount of upward motion. The middle cluster is moving rightward, as we see. This E1 component is rightward. And the third cluster is moving a little bit leftward, not so sure. So we can do this, okay? But the point that we realized here was already that, this is a problem. These cells are not really behaving in a mean field manner. We need to do something different. We maybe need to really start thinking about them more as individuals, right? So we need to go to this idea that these are individual agents. And so we're asking, okay, can we somehow go beyond this mean field response and look at cells as agents? That's what we're talking about. This is where we get into this idea of Markov decision processes and so forth, okay? So we say, okay, let's now look at this whole thing as each cell being an agent that is undergoing these Markov decision processes. A little bit formal, a little more formal here. So we look at each cell here as an agent. Each cell is an agent. It is in some environment, it could be the body. The cell has certain states, and we'll say what these states are. Right? From each state, this agent, the cell agent, can take an action, right? Change to another state. So they follow dynamics. Can be regarded as Markov decision process. They have some states. They're taking some actions. There's some initial probability of being in a certain state. So P is the probability. There's a transition probability, which says that, okay, if you're in some state S, and you take some action A, what is the probability that you're going to end up in some state S prime? This is a transition probability T. Now, the thing that goes into our treatment here for this particular MDP, Markov decision process, is to say that, well, maybe we can find a reward function, right? So are the cells trying to optimize something? Are they trying to maximize or minimize something? We say maximize, we call it a reward, okay? So we say, okay, maybe we can find something that we can identify as a reward for them to being in some state and taking some action. Now there's more machinery here. You observe the dynamics here, and there is this notion of a policy. A policy is also it's a probability distribution. The policy is the probability of taking a certain action if you are in a certain state, right? So you find yourself in a certain state, you take an action. What's the probability of taking that action? That's the policy. There's a notion of the optimal policy. The optimal policy is something that maximizes a cumulative reward and the expected value of the cumulative reward. The cumulative reward is to say that I don't care only about the reward I'm going to get right now, but what is the reward I'm likely to get over time, right? Or as I go through very many different states. So this would be that would be the optimal policy for you to follow, right? So don't be greedy, just look for your reward now, but think ahead. Okay. The last thing that we also need is something called a state action value function. It's a similar idea. It is to say that you take the current value of the reward you get, plus what do you expect for your future, right? So kind of save for the future. And that value is another important idea here. Okay. The whole idea, and like I said earlier, I'll just reiterate, the reward is the motivation, like driving the agent behavior, saying right now what you're going to get. The policy and transition are probability functions which together describe these dynamics. Okay. Right, so some amount of formalism here. We'll be using this. Okay. Now, when one looks at MDP a little more here, this is where learning comes in, right? So say, okay, if I see a certain, if I have trajectories, right? I have a Markov decision process where I have these agents, cells, occupying certain states. They can take certain actions. There's an initial probability of their distribution. Maybe I have a transition function to know how they're going to change between one state, given a state and action. What is the probability they'll be in another state? And there's some idea of reward. Now, reinforcement learning algorithms take all of this information and try to find the optimal policy that the cells are following. There's a big diagram here. It basically says that you have certain trajectories, you have some initial probability. The trajectories go from 0 to final time t. You're also taking actions from between an initial time and a final time. And then there's this notion of transition and reward, and all of this is used to determine the policy. This is standard reinforcement learning, where the central quantity you're trying to get is this policy, and this is a probability distribution function. You also want the value, but the policy is really central. It tells you in a state, what is the probability that you will take a certain action? Okay. But here, what we observe is that, okay, for reinforcement learning, you need to know the transition. You need to know what the reward is. Here, we don't know this for cells. So we need to do something else. We need to also figure out what the reward is. And actually, what we need to have some insight to the transition also. What this means is, and we also want to figure out the policy. So really, we are solving an inverse problem. This brings us to inverse reinforcement learning. Now, conventional inverse reinforcement learning, and we use the acronym IRL, it says given trajectories and a transition function, you try to infer the reward, right? So I won't go through all these details here. I'll just go to the diagram. Again, you're given trajectories. Those are the data, right? They start out with some initial probability of being in some state. And essentially with this inverse reinforcement learning, you can go from trajectories directly to transitions, or you may have some knowledge about what the transitions are. Traditional inverse reinforcement learning algorithms just look at the trajectories and empirically say, okay, I can see how these agents are doing all these things. I can see how these cells are changing states. I can simply empirically, just by counting, figure out a transition probability. Okay, that's an empirical one. The problem with doing this is that it's poor at extrapolation because you're just going with the data that you have. You're just seeing the system that you have. Extrapolation is going to be bad, as we know all these processes, right? All these sort of data-driven processes. So the question is, can we introduce some mechanistic behavior or can we introduce some physics to this, right? Can we have a more principled way of coming up with these transitions? Once we have that, we can think about IRL algorithms, which also infer the reward and the policy, right? So remember, all of this we're going to bring to bear upon cells. How do we do this? We took two approaches. I'll go through the first approach rather quickly because it's not, well, it was our first shot at it. Remember, we said that the cells can be in 3 clusters. At the outset, we said, okay, let's look at the cell initially, right at the outset, choosing to belong to one of these clusters, right? Choosing to say that I want to be one of these guys moving left or right, or I'm not so sure. That we said initially is an action. So we had effectively a discrete action space, right? The cell could take one of 3 choices and it had to make them at the beginning. Okay. All right. So with that idea, the approach we took to solving this inverse reinforcement learning problem is one where we said, okay, we use algorithms which would maximize the total reward, the cumulative reward over all times, while also accounting for the possibility of also concurrently saying that, look, I don't know what the future holds, so I'm going to maximize the uncertainty of my prediction. In particular, there's a quantity called causal entropy. These type of IRL algorithms were described more than 10 years ago. That's called maximizing causal entropy, right? So that's what these algorithms do. But still remember, what about the transition probability, right? It needs a transition probability function. So this is where we said, oh, wait, I know how to, if I look at these data, I can go back to my inference methods, my system identification methods, and maybe from there I can get something. What can I get here? Let's look at the right part of the slide here. It turns out that the cell concentrations, when rescaled, can be actually regarded as a probability density function, right? You look at the concentration of cells somewhere, you can turn that into a probability density function. And then we have available to us the Fokker-Planck equation, right? Which essentially, I'm continuing to use C here, but think about C as a probability density function. Okay, so in the Fokker-Planck equation, psi here is a potential, and essentially you're looking at the gradient of a potential, and you have this divergence coming from the gradient of a potential and another diffusion term. Of course, the gradient of a potential, if you just stick in a constant vector for it, gives you back advection. Okay, but this form is the more general one, which is the Fokker-Planck equation. There's been a lot of work on it, and a few seminal papers coming out from these from Jordan Kinderlehrer and Otto. In '98, they showed that this form, the Fokker-Planck equation, is a Wasserstein gradient flow of the entropy regularized energy. What that means essentially is that the system is minimizing this total free energy consisting of a potential. Now we have a potential for the cells. It is a potential coming from that chemoattractant, right, which is a chemical gradient there. And then there's an entropy term. Now, we realized then that, hang on, we are minimizing this quantity that's equivalent to maximizing the negative of this quantity. We said, can we connect this maximization of the negative potential with the fact that we are maximizing a total reward in these reinforcement learning or in these MDP processes? So as a sort of conjecture, we said, okay, maybe I can determine the potential for these 3 cell types, these 3 populations, and use that to write my reward function, right? So because I needed to have some sense of what this reward function could be. I still need to determine the coefficients, but I still could use the potential for each of these 3. If I can identify a potential for each of these 3 cell groups, I could say, okay, I can expand my reward in those terms. We could do that. And then we go ahead and do our inference method, the system identification methods I showed you earlier. For the same cell group, I think I showed you these values that we got earlier, but now we're reinterpreting them as a potential. So psi for the three groups is shown here. S1, S2, okay. S1, S2 are basically positions. You can think about them as the X and Y coordinates. So the state here we're seeing is position. So this is the potential for the first group, the potential for the second group, and the potential for the third group. Now, because we've identified Fokker-Planck equations for each of them. We also get from the Fokker-Planck equation, the Fokker-Planck equation can be connected to a discrete probabilistic form also. That gives us a transition probability, which is in this form. If you look at this carefully, you may recognize actually the fundamental solution of the heat equation also sitting inside it, right? It has relations to all this. That allows us to write out the reward function in this fashion, right? So for each cell group, I have determined what the potential is, psi 0, psi 1, and psi 2 for the three groups. And then I have to determine these three quantities, my thetas. If I can infer all of that, I have a reward. Okay? And we described this approach in a paper. Okay, so what do we find? This part is a little abstract. I'm going to go over it quickly, but okay, we find this is what the reward looks like for the three cell groups. This is position, X direction, Y direction. For all three cell groups where the reward is warmer, that's where they should be going, right? So the method realizes that's where they should be going. But interestingly, it finds that even this middle group, this is the group of cells which are moving rightward. They know the reward is to the left, but for some reason they're moving rightward. So the method kind of the IRL algorithm we have here recognizes that these cells are moving against the imposed gradient. That's this group here. How good is our inference here with IRL? We are finding these are the policies, right? So the policies are what is the probability of you taking a certain action when you're in a given state. What it is saying is that for group one, if they are toward the left end, their probability of moving leftward is higher. For the group in the middle, if this is the group that's moving rightward, it does recognize that you already are rightward. That's what makes you go in the direction that you're going. And this is the actual data for it, right? So we find policies that kind of connect up well with what the cells are really doing. However, this treatment was not very satisfactory. It was not satisfactory mainly because we were already sort of forcing the cells to choose what their actions were. The cells had to decide at the beginning whether they wanted to be group one moving left, group two moving right, or group three, not quite sure. And then even what we found was really just reflecting what we had chosen. So not a very suitable approach. So we said, okay, we really have to go away from this idea of pre-deciding that you have only three actions to choose from, making action and a discrete space and like forcing you to be fixed there. So we had to describe a different approach. We had to come up with a different approach which would allow the cells to choose from continuous actions and also on the fly decide to take different actions from the ones they've taken in the past, right? To sort of change their minds. What this meant formally was that we had to put state and action into a continuous space, a single continuous state action space. So we call this X. Now X refers to this joint space of states and actions. Turns out that even this type of behavior can be modeled by a multivariable Fokker-Planck equation for the time evolution of the probability density, which now depends on X, where X, however, is your state and occupation, state and action. The Fokker-Planck equation looks the same, except that now all of these X's, instead of being only position, are like, a state and action. Okay. If you and again, the joint probability transition probability function is also as before, as when you can solve the Fokker-Planck equation, you can get it as a function of the potential that you may infer for this multivariate Fokker-Planck equation. Okay. Looking at the Fokker-Planck equation and the MDP, we recognize that there is an isomorphism in structure between the Fokker-Planck equation and these MDPs. Specifically, we observed that the potential function in the Fokker-Planck equation is equivalent to this other quantity that I introduced. It's called the state action value function. Just tell you again what it is. It is what the reward that you have presently plus the reward that you expect to get over all the possible trajectories you may take in the future. Okay? We found this kind of isomorphism between them. Structurally, they seem to be playing exactly the same role. Why? In the Fokker-Planck equation, the potential drives the lumped state evolution in the Fokker-Planck dynamics. Right? It's the potential which is the driving. In MDPs, it is again the state action value function that sort of guides the agent's actions and therefore states. We found that if we made this identification, a bunch of other things flow. Also, I apologize, this looks heavy, but I'll tell you what each case is doing. From Fokker-Planck dynamics, it turns out that the steady state distribution, you can show it that it is the minimizer of the free energy. Okay? And this is a result that one can derive from Fokker-Planck. So it says that this distribution is the minimizer of the energy, and it has this particular form, right? This comes from the Fokker-Planck equation. Now, this depends upon states and actions. One can marginalize this over the actions by just doing this integration over action space. So now you have the steady state marginal distribution only in terms of states. The conditional distribution then of an action is given by the ratio of what you have in this first line and the second line, right? It's this, takes on this form. This structure here has the same form as the Boltzmann policy from reinforcement learning, okay? And why there's a further connection here. It says this quantity here from Fokker-Planck says, what is the probability of you having taken an action from a given state at steady state? This tells you, again, the Boltzmann policy, right? What is the probability of taking an action from a given state, right? So they're even conceptually, they're the same thing. And we observe that their structure is also it is actually identical. So this is kind of evidence sort of supporting our conjecture. It's still a conjecture. It's not a theorem. Okay. So we have this connection. Furthermore, we can get a joint transition function from the Fokker-Planck. Okay. With this transition function, one can and the state value action function, which is equal to the policy, which is equal to the potential from Fokker-Planck, one can obtain the reward through an equation called the inverse Bellman equation in MDPs. We observed a theorem which is basically saying that there is a one-to-one, that there's a bijective map between a value function and the reward. Okay? That's just the theorem. It's not important. It just says that if you have a value function, you can get a reward from it. Okay. What do we do with this? So here's our approach. We say, okay, you infer a Fokker-Planck equation. Okay, given these data, you need to parameterize a potential function. We parameterize it in terms of how many polynomials. We do our system identification for Fokker-Planck. Okay, we get some diffusivity and we get some other parameters, right? So we do this. With that, since we know the potential function from Fokker-Planck, we observe that the potential function, once we have the potential function, that potential function is the same as the state action value function for the Markov decision process. That gives us the Boltzmann policy for the Markov process by this observation that we made from our conjecture. We get a joint transition probability function for this cell that is in state S, takes action A to go to state S prime with action A prime. We can also marginalize that transition probability function to get now the probability of just being in a certain state given a state in action. Finally, we can use this thing called the inverse Bellman equation to come up with the reward. So the whole idea is that if you can look at the cells, come up with the Fokker-Planck equation, that actually gives us a lot of information to solve this MDP, which defines the cell's evolutions as agents. Okay. And I'm running out of time here, so I'll just show you some results we got to this. We first set up a synthetic problem. The details of the synthetic problem are not how we set it up is not important. These are not cells, right? We just created some data, let the system evolve, and then applied our combined approach. So now we have an approach, if you recognize, where we can solve a Fokker-Planck equation and use it to drive this inverse reinforcement learning. So we call this Fokker-Planck inverse reinforcement learning. This is the approach we apply to our data. On the left here, we see the ground truth value function. The X and Y axes here are states. Okay, so these are positions in this case. And we also have the action, right? This is a joint space. So within each of these squares, if you see each of these squares is also colored. So the colors within the squares give you the actions, right? So because there's actually a four-dimensional space. So I have to have some way of showing this. So I'm showing you 2D, and inside each of these squares for a given values of states, we're showing you the actions. So this is our way of showing this four-dimensional space. This is again showing the reward. This just shows for our synthetic data how the probability density is evolving. It starts at time t equals 0, goes as far as time t equals 19. So these states are evolving towards these two hotspots here, right? So as the thing proceeds. We applied our Fokker-Planck inverse reinforcement learning to this. So we generated trajectories, we solved the Fokker-Planck equation for it, and then sort of tried to recover what we'd originally posed as ground truth. And this is what we got, right? Up here, you see the ground truth reward. This is the inferred reward below, not a bad match, sorry. This is the ground truth value function. This is the inferred value function from a Fokker-Planck inverse reinforcement learning. This is a plot showing the KL divergence, which is KL divergence between the data and the inferred probability distributions, right? Also declining. As it declines, it means those two distributions are coming closer together. And this just shows a sort of L2 error for state and action. Okay, as we refined the mesh, as we went to more and more states, the states are defined over the same space, but as we went to more and more finer grid, we're seeing that the error goes up. So this is a synthetic problem. Of course, we want to apply it to this problem, right? This cancer cell dynamics. So same thing. Now I'm coming back to those cells. In addition to moving around, when I showed you many movies, the cells moving around, these cells are also expressing quantities, right? They're expressing many kinases and proteins and so on. Two in particular that our biologist collaborators focused on are AKT and ERK. We don't need to get into what exactly they are, but they're what are called kinase transpose reporters. Both these quantities, these proteins AKT and ERK, are their kinases, are related to motion in various ways. Right? There's a correlation between them. So this again is a kymograph seeing about thousands, of the order of about 1,000 cells going up here. Each of these traces shows how the cells are expressing AKT over time. So in each of these plots, the horizontal axis from here to here is time, and these are different cells. So that is their AKT expression, this is the ERK expression, this is the speed, and this is their directional speed, right? So this is what our biologists are observing. With these data, we said, okay, we will apply it to our Fokker-Planck inverse reinforcement learning. Now we have to define what states are. Unlike earlier when we were using positions as states, we decided here to use velocities. There's a lot of detail about why we came up with velocities. So the states are the velocities of these cells. The actions, now we'd made a difference here, right? We said the actions, instead of initially what we said was that the actions were, you had to choose which group you wanted to belong in. Now we're saying the actions are a continuous space, right? And they're just what is your expression level of these two kinases of ERK and AKT. Those are our actions. So with our Fokker-Planck inverse reinforcement learning, we wanted to see is, okay, is there a relation between these velocities and the expression of these kinases? So we applied our approach, determined the Fokker-Planck equation for these quantities, used that to solve the IRL problem. This is what we see. Again, very abstract plots, not at all clear what's going on here, but let me tell you. This is a plot of the reward. So the colors, wherever it's hotter, is where you get a bigger reward, right? The cells are getting a higher reward here. The axes X and Y are states. So they show velocities. So everything is normalized, right? So in the middle, you have a zero velocity. You have a positive velocity to the right at this end here, velocity to the left here, up and down along the y-axis, okay? So if you look at this, we see immediately, okay, you get more reward if you're moving leftward and toward the top, right? So this is again in the direction of where we know the reward is, right? Because the gradient goes to the left, so the cells moving to the left indeed get a higher reward. We are seeing that. But here's the other interesting thing. Hopefully you can see these tiny squares here. Inside each of those squares, each square now is a particular value of X and Y velocity, right? So let's look at one of these squares. Inside that square, hopefully you can see smaller squares if you're able to stare at your screen. Those smaller squares give us the expression level of these two kinases at that combination of velocities. So we look at where the rewards are high and we see, okay, the highest reward within any of these squares is where we get the yellowest value. But that is obtained where, if you look at the square, you will see that on the horizontal axis within the square, you get a higher reward if you're slightly to the left, and along the vertical axis of the square if you're higher up. So what that translates to in terms of these two kinases we're looking at is low values of AKT. AKT is plotted along the horizontal axis in each square, and you've got to be to the left in each square. So that's low AKT. And you need to be toward the top in each square, that's high ERG. This is what our methods inferred. This shows the policy, and I'm not going to get into too much. Let's just look at the reward. So we went to our biologist friends and told them this, and then they realized that actually okay. So they said, actually, this is what we've been doing to these cells. The same cells, they've applied a number of different interventions to them, right? They've been doing a lot of chemical interventions to them. This is the control, no chemicals applied, bunch of different chemicals. This is an interesting one. They've applied trametinib. This is a chemical that actually knocks down the action of ERK, which is one of these kinases. So when ERK is knocked down, the velocities become very small, right? Compared with the control. These are trajectories, right? So as you can see, the trajectories are much more limited. They're not moving that much. Okay. The one on the right is alpesolib, which is another chemical which actually knocks down the action of AKT, the other kinase. Interestingly, when they knock down the action of AKT, the trajectories are a little bigger than the control, right? If you look at this, on average, they're a little bigger. They did a more careful analysis of this, and indeed they found that when they knock this down, they get a slightly greater velocity when they put down AKT. So essentially what we're finding is that when we, from our prediction, which said that AKT should be low and ERK should be high, that is exactly what they realized after the fact. This was blind to us. We reported this to them, and then they came up with the fact that they had run these controls and found these results. There's this idea that theory only takes you so far. I expanded that to what Oppenheimer is supposed to have said. He said, well, they won't fear it until they understand it. They won't understand it until they've used it. So we showed it to the biologists and said, well, this is how all these methods, these AI methods can be useful, actually showing you something. That's it, really. There's just details about the algorithms. So to summarize, you can use mean field approaches to describe cell dynamics. It breaks down. We're interested in heterogeneity, and we've gone to this approach where we can connect Fokker-Planck with inverse reinforcement learning. There is this conjecture that there is a connection between them. It's something that we're still working on, trying to sort of beef it up, but we're definitely seeing a lot of evidence that suggests that this may make sense. As a preprint out, and there are many greater opportunities in looking at optimal transportation theory. The Fokker-Planck equation, as many of you may know very well, is part of optimal transportation theory. Our work has been supported by Keck, and these are all the people. So everybody in the top line is either grad students or postdocs who've been doing all the work. Changyang Huang and Siddhartha Srivastava and also Xun Huan did a lot of this work. Patrick Kinnan, and also Kenneth Ho is the postdoc who did all the experimental stuff. All my colleagues, the Lukers are biologists. Nicola Banovich and Xun Huan are data scientists. Xun Huan, many of you may know, was at Sandia Labs. And yeah, that's where I should end here.
Awesome.
Didn't go too far.
Awesome. Thank you so much. Wonderful talk. Pretty informal. I was able to follow. Not all of them. Not all of them, but
we are also still learning. I got to confess, we're learning this also in many ways. On both sides, even the AI, even the IRL stuff, we're learning.
Great. Okay, thank you so much. Let's have a Q&A session. If you have any question, please unmute yourself and ask your question. So you're welcome to do so now, because it's a Q&A. I can start. You basically show, in gradually and in procedural way. I mean, you show like, let's divide it to 3 groups and then, conquer separately and then combine them later. And that approach, and later on, using the conjecture you have between the isomorphism and the Fokker-Planck and, Markov chain process, you get rid of those, the dividing process and just monolithically solve it.
Exactly.
Is there a way of, comparing those two methods and find out the performance between those two and measuring the...
Right. So it turns out that the descriptions are so different that they almost become apples and oranges, because the first approach, because with the first approach, the way we are fixing actions, the actions are no longer available for us to evaluate, right? We fix them. Furthermore, we're even saying that the cells have to decide at the very beginning what they want to be, so they cannot switch around. So of course, we could compare, we could look at maybe the L2 error in our predictions, right? Though in the second case, our space is bigger, so maybe we take the subspace of just maybe just look at the subspace of velocities and compare with what velocities we were getting in the other case. That's a possibility. Even that becomes difficult, because in the first case, the velocities were just 3 velocities, right? For each. So the comparison is a little difficult in that sense. We could look at the reward, I guess. We could look at the reward for a synthetic problem. That is something we could do, right?
I see.
So we did not do that. So we would need to set up a synthetic problem, approach it the 2 different ways, and try to come up with them.
In terms of computational cost, I guess the latter one is more demanding computation.
Much more demanding computation.
The first one.
The first one is much quicker. The second one is more demanding because we are solving in a 4-dimensional space, so it scales as we refine. Our description itself is 4D, so it explodes pretty quickly.
Right.
But on GPUs, it's and then the IRL also for the same reason. But on GPUs, we can get some benefit. We are sort of we went up to about a 20 by 20 grid for all 4 space, all 4 dimensions, and that was sort of on the edge of what we could get on CPUs with even with I think of the order of tens of nodes, maybe 20 nodes on Expanse. We were getting, it was still taking a few hours for each case. So but there's some acceleration, but it's expensive. Yes,
I see. Well, it's a fascinating and I think the new idea you're exploring right now. Hopefully it'll take you to the really groundbreaking.
Well, hopefully we'll see. It's nice because there's some theory and there are some results, some applications also.
Right. If you don't mind, like going to slide 7, very beginning of your talk, I saw that inferred the results is there's just some I can almost see some vertical stripes.
Exactly.
Is it normal? Is it supposed to be?
No, I think that's just a function of what time steps we were using for the. I suspect that in this case, the time steps we were using for the inference were larger than what we were.
I see.
Than we had from the data in this particular case.
I see. I guess
I think that's
some numerical.
That's a numerical artifact.
I see. And then you talked about the variational, the system identification.
Yes.
Kind of like the weak form of the Cindy. Very, very interesting. You know the groups in Colorado at Boulder, they come up with a weak Cindy as well.
The weak Cindy. So they followed ours. They came out just after our papers had appeared. They got in touch with us, and they were describing these methods. We're aware of those methods. We have not. We haven't pursued the system identification itself much more in that direction.
Yeah.
We've gone off in other directions in terms of applying it to a bunch of other problems and just trying to come up with better ways to do this. We're also looking at some we looked a little bit at genetic algorithms to sort of make the choices.
Yeah.
It turns out that this problem of choosing these operators or which ones to turn on and off, it can become combinatorially very large very quickly because actually if you look at the graph of all possible models, it can get very, very big. So one possibility we're looking there is to see whether these methods themselves, because of their combinatorial growth, become interesting for quantum computing. Whether this sort of approach is because if you want to truly explore all possible models, we follow the greedy approach. If you want to truly explore all models, that becomes a very, very, very big problem.
Right.
Very quickly. So that's the direction. But sorry, you may have had a question on weak sending.
Oh yeah, I mean, I as a matter of fact, I do work with them.
Oh, I see.
And then one of the key features they always highlight is like whenever there is a noise in the data,
the weak form
it's so much more robust than strong form of the Cindy.
That's right.
And I guess you answer my expected questions by saying that you kind of went to different route. You didn't go into the route of the identification realm much.
Right. No, we have not followed up the questions of how do you characterize the amount of noise that these things that these weak approaches would still work well with. We haven't gone down. We haven't gone down that route.
I see. All right. I guess I'm asking too many questions. I have to give a chance to audiences to ask questions. If you have any question, please unmute yourself and ask. All right.
End of the week.
One last question for me.
Yes, please.
I was saving one. If you go to slide 4, like previous slide, you are kind of modeling the diffusion terms and advection velocity terms with some sort of polynomials. And why up to only 4th order? You could go to 6th order.
We could just keep going, but
keep going.
That's the thing with all these sparse regression approaches, right? I mean, ultimately they're regression approaches, so we always start out with a library, and at some point you could you put an end to the how far you're going out with the library.
Right.
We could go.
I guess you haven't explored those things.
We haven't gone that far. I mean, in this case, we were quite driven by wanting to come up with something that was explaining the question, the phenomenon well enough, and then proceed on to other things here. We could have gone on with
yeah. All right.
Okay, great. Thanks so much.
Thank you so much for the wonderful talk, and let's thank our speaker today.
Yes, if I may, thank you all very, very much. If I may have your attention, hold you for just a second.
Yeah.
So I'm changing coordinates, and beginning of January, I'll be moving to southern USC.
Oh, you are?
Yeah,
it's closer to us now.
Yeah, much closer, right? Just down the coast. And one of the really interesting things at USC is that there is this big initiative in computing, in advanced computing. It's called, so if you ever have a chance, just look at computing.usc.edu. There is lots of hiring is going to be going on here at senior and intermediate levels in computational science of the kind we all do. So keep an eye open for it.
I guess you're kind of leading that,
Well, I'll have a role. There are aspects of it, yeah.
I'm sure you're going to play an important role there. Well, thank you for letting us know. It's
Definitely, yeah.
exciting. Yes.
Yep.
Yeah. Okay.
Okay. Well,
Well,
great.
thank you for
Thanks
the
so much.
wonderful talk. Yeah.
Thank you.
Thank you.
Thanks for the invitation. Thanks, everybody.
Thank you.
Yep.
Krishna?
Yeah.